In the series of videos, we have been discussing about the cellular respiration, where we discussed about glycolysis first and then we discussed about citric acid cycle, where we saw the formation of some ATPs and also there was reduction of some molecules like NAD plus and FADH molecules. The reduced form of NAD plus and FADH are NADH and FADH2 and are called electron carriers, as they will deliver their accepted electrons to the electron transport chain that drives in the inner mitochondrial membrane. The electrons are transitioned between complex molecules, and in that they lose energy which is used to pump protons from matrix to the intermembrane space, and ultimately we will get the production of ATPs via chemiosmosis. This will be explained in a detailed manner in the upcoming video. So here we will be more focused towards the energetics of glycolysis and citric acid cycle. We often get confused to the total net production of ATPs from these processes. First of all, let's see what glycolysis gives us. From glycolysis we get 2 ATPs and 2 NADH molecules. And from 2 turns of citric acid cycle, because you know this citric acid cycle runs 2 times to oxidize the 2 molecules of pyruvate. So in these 2 turns we get 2 GTP molecules, 8 NADH and 2 FADH2 molecules. And before getting to the calculation part, remember that 1 NADH molecule yields 2.5 ATPs in oxidative phosphorylation via electron transport chain. So the question is, why 1 NADH yields only 2.5 electrons, not 2, not 3, why 2.5? When we will see the electron transport chain, we find that complete oxidation of 1 molecule of NADH pumps 10 protons out of matrix. It must be noted here, its 10 protons are pumped out of the matrix. And you know it's the passing of 4 protons through ATP synthase to yield 1 ATP molecule. That takes us to the conclusion, 10 protons divided by 4 protons will yield us 2.5 ATPs. And in the same way, 1 FADH2 molecule will yield 1.5 ATPs since it pumps 6 protons out of the matrix. That's 6 divided by 4 gives us 1.5 ATPs. This is the calculation part of NADH and FADH yield. Now first of all, let's see the energetics of Kreb cycle first. From 2 turns of Kreb cycle, we get 8 NADH molecules. And this 8 NADH molecule is to be multiplied with 2.5 ATPs. That is the value of 1 NADH molecule in oxidative phosphorylation. So in total we will get 20 ATPs. 8 multiplied by 2.5. That means we will get 20 ATPs from 8 NADH molecules. Then also we have 2 FADH molecules being produced in 2 turns of Kreb cycle, which is to be multiplied with 1.5 ATPs. That is the value of 1 FADH molecule in oxidative phosphorylation. And from this we will get 3 ATPs. That's 2 multiplied by 1.5 ATPs. And also we have 2 GTPs. That's 1 GTP is being produced in 1 turn of Kreb cycle. That means we will get 2 GTPs and they are somewhat equal to 2 ATPs. So this is the energetics of Kreb cycle. Now let's have a look at glycolysis. First of all, we have 2 ATPs directly produced from glycolysis. Then we have 2 NADH molecules. These NADH molecules are very different from the Kreb cycle NADH molecules, since these NADH are produced in cytosol, because this glycolysis runs in cytosol. So in order to implement their electrons in electron transport chain, the electrons from these carriers need to be shuttled into the inner mitochondrial membrane for oxidative phosphorylation. Here we see 2 different shuttle mechanisms are present in order to carry electrons from NADH molecule to the electron transport chain for oxidative phosphorylation. We have malate aspartate shuttle and glycerol phosphate shuttle. Now what's the difference? If electrons from NADH are delivered via malate shuttle, then 2.5 value remains the same for NADH. That's 1 NADH will yield 2.5 ATPs, because in this shuttle NADH is regenerated in the matrix of mitochondria. Whereas if the electrons from NADH are delivered via glycerol shuttle, then value decreases to 1.5 ATPs per NADH molecule. As in this glycerol phosphate shuttle, the final regenerated molecule in matrix is FADH2 instead of NADH, which is first oxidized. So the value comes out to be 1.5 ATPs from glycerol phosphate shuttle. Now it depends which shuttle is used in the oxidative phosphorylation, and due to that either we get 3 ATPs or 5 ATPs from 2 NADH molecules produced from glycolysis. So when we take out the total calculation, or we can say total net gain of ATPs in the cellular respiration, it's 30 to 32 ATPs. The range is only due to the presence of two different shuttle mechanisms, malate and glycerol phosphate shuttle. So this is all about calculation, or we can say energetics of Krebs cycle and glycolysis. I hope you like the video. If you like it, give it a thumbs up and make sure to subscribe this channel. Thanks.